Welcome to the third lecture in Bio 110 about cytoskeletons. Today we're going to be looking at intermediate filaments as well as some of the cytoskeletal functions that these molecules produce. So today we're going to be looking at the intermediate filament, and as is shown here in the left, the intermediate filaments are a type of rope-like molecule that can span between different cells. Let's take a look at those cells more carefully. Intermediate filaments. They're very strong and they're rope-like. In a sense, they're similar to actin filaments in muscle. And they are, however, not used by any motor proteins that we know. So they're not used to transport organelles just like actin and microtubule filaments are. Now, let me introduce you to this little film first so you can get an idea about their architecture and biology, and then we can go a little bit more in depth about it.
Eukaryotic cells contain a complex network of filaments, intermediate filaments, microtubules, and actin filaments, that provide the cells with strength, structure, and movement. Although all eukaryotic cells contain microtubules and actin filaments, intermediate filaments are found only in vertebrates and a number of other soft-bodied animals. Intermediate filaments are found in animal cells that require a lot of strength, such as the epithelial cells of the skin. Some of these filaments span the length of the cell, connecting cell-cell junctions called desmosomes. These cables of intermediate filaments have a high tensile strength. Without these filaments, stretching or pressure on the epithelial sheet would cause it to rupture. Each filament is rope-like, consisting of 8 thinner strands made of a precise hierarchical arrangement of protein subunits. At the lowest level, 2 monomers associate with each other to create a twisted dimer. 2 dimers then line up to form a staggered tetramer. Note that the 2 dimers are arranged in opposite orientations, with their amino terminal ends away from each other, so that the 2 ends of the tetramer are indistinguishable. Tetramers then link end to end, thus building up one strand of an intermediate filament. A total of 8 strands stack together and twist around each other to create the intermediate filament. This stacking provides the extensive lateral contacts between the strands that give the filament its remarkable mechanical strength. An electron micrograph shows the appearance of intermediate filaments that have been assembled in a test tube.
All right. Let's take a look at this in more detail. So as the movie indicated, an intermediate filament, it's made of an alpha helical monomer. That monomer has an amino group and a carboxyl group. 2 alpha helical monomers will become a coil-coil dimer. And again, take a moment to look at chapter 4 for you to remember what a coil-coil dimer is. Now, you have one molecule in which the amino groups are together, and one molecule in the other side you have the carboxyl groups together. 2 of these dimers, 2 of these coil-coil dimers will then come together and form the staggered tetramer. And as you can see, you have an amino group and a carboxyl group in one side, and an amino group and a carboxyl group in the other. And that is why these monomers are indistinguishable. The sites are the same. So now, 8 of these molecules will associate laterally to form a tetramer, and then the tetramers will come on together to form the very long filaments. So if you take a look at this molecule more carefully, what you will see is that the intermediate filaments are very thin but very, very strong. Their tensile strength is high, they're tough, and they're durable, and because of that they're able to withstand a large amount of mechanical stress. Now, in the metal, if you were to break down an intermediate filament, you will have 32 individual alpha helical coils. Therefore, each of these coils gives its independent strength to the filament, making it very strong. What we have, this is an analogy for a cable. Here in the left, you can see a cable that is used in the Golden Gate Bridge. But when you cut that cable across and you want to see what is inside, each of those cables, it's made of independent filaments that give us a lot of strength. So therefore, the entire cable composed of 7 different smaller cables, each is made of a smaller cable. So this is how in a similar fashion, this is how this cable is able to be very strong, and therefore, in a similar way, the intermediate filaments have a lot of strength. Now, we're going to look at the family of intermediate filaments. We can divide them into 2 types. The nuclear intermediate filaments are going to be the nuclear lamins, and those are present in all cells. They are the molecules that are going to give the structure to the nucleus. But you also have cytoplasmic intermediate filaments, and those are going to change depending on the cell. We're going to be looking at epithelial cells, and there you're going to have keratin. So there's going to be very different types of keratin, and each of those keratins are usually expressed in a particular epithelial cell tissue. We're going to look at connective tissue, muscles, and glial cells, and on there you're going to have vimentin and vimentin-related filaments. Last but not least, in nerve cells you have neurofilaments, and all of them are required to provide the cell with structure to sustain stress. Now, the intermediate filaments are going to add strength to the cell, allowing it to withstand a lot of mechanical strength. So here in the left, what we have is a cell that has the intermediate filaments, and as is shown in this cell, the intermediate filaments are connected from cell to cell by structure called the desmosomes. In this, hello kitty. In this picture, what you also see is that in this epithelial layer, what you have is a basal lamina, shown here in yellow, and the intermediate filaments bound to the basal lamina are aggregating over there at the structure called a hemidesmosome. On the right, you have an epithelial layer that doesn't have intermediate filaments. So when that cell, that epithelial tissue becomes to endure stress, mechanical stress, the intermediate filaments inside that are connecting all the cell walls between epithelial cells, all around the lateral side and the basal side of the epithelial layer, are able to provide that cell with a lot of strength, versus the cell on the right, that epithelial layer without intermediate filaments, it's not able to withstand the shear. So now let's take a quick look at some of the epithelial layers. And in here in the left, what we have is a fluorescent microscopy image of epithelial cells containing keratin. And keratin is the intermediate filament that is present in epithelial cells. What you can appreciate on the right is the fact that this epithelial cell has areas that are joining with the membrane of other cells, and those are going to be the desmosomes. You can see that the intermediate filament bundles at the area of the desmosomes, and the desmosomes will connect to different cells. In number, all the epithelial cells are joined together by desmosomes, and therefore the desmosomes serve as a point to reinforce the cell with the intermediate filaments. As I mentioned, the hemidesmosomes are the way in which the intermediate filaments are attached to the extracellular matrix, usually at the basal lamina. So now, defects in epithelial intermediate filaments, in this case a defect in keratin 5 and keratin 14, are responsible for a condition called epidermolysis bullosa simplex. Bullosa means blisters, lysis you mean breakdown, and epidermis, as you know, it's the outer skin of the cell. So in this rare genetic condition, the epidermis is not able to withstand mechanical stress, and therefore, if applied to it, it will detach from the dermis, creating the blisters that are shown on the right in this small baby. So usually people with this rare skin disorder have to wear reinforcement on their skin to allow it not to fall apart, and therefore they could then be prone to infections and have severe discomfort and issues. But as you can see from this image in the bottom part, the epidermis, it's not able to withstand the shear stress and therefore separates from the dermis, generating the blisters that are going to be characteristic of epidermolysis of Epidermolysis bullosa simplex. Now, other intermediate filaments can be cross-linked together to microtubules by a protein called plectin. So plectin, it's a molecule shown here in yellow, and the green, the blue fibers are the intermediate filaments. In between them, you have connected fibers, these are proteins that are going to connect to the red microtubules shown here in the image. So in that way, the microtubule network and the intermediate filament network are connected by molecules like plectin, therefore providing a lot of structure to the cell and also dynamic sensitivity as you know, the microtubule network is able to grow and compact itself. In neural cells, what we're going to see are the presence of neurofilaments, and neurofilaments are going to be the intermediate filaments in neural cells that are going to provide them with strength. So we have here three images, one of the axon, one of the glia, and then a cross across the axon showing the intermediate filaments and the macrotubules. So we can see three different types of neurofilaments: neurofilament L, neurofilament M, and neurofilament H. Those are different in their architecture. The neurofilament M and H have very long C-terminal tails, and those are going to allow them to bind to neighboring filaments. That is going to provide you with really uniform spacing between the macrofilaments. So when the axon is growing, the subunits are added to the filament to make sure that it extends and they reach to the end. Now, what you can appreciate from the right, it's the very regular spacings of the neurofilaments in the axon of the cell. The axons are going to be populated with a lot of little intermediate filaments shown here as small black dots, and along with it, what you can appreciate are the circles, and those circles are microtubules who have been cut across, and therefore you can see that the intermediate filament network, it's dotted with the macrotubule network that allows, for example, for the movement of vesicles from the cell body to the axon terminal. One problem that we understand that can happen when we have an abnormal accumulations in macrofilaments in a motor neuron cell, it's exemplified in amyotrophic lateral sclerosis, or the Lou Gehrig disease. Probably you have heard about the Ice Bucket Challenge, and this is the disease that the Ice Bucket Challenge was trying to address. So, because there is problems with the neurofilaments, and they are accumulated abnormally, those neurons now do not have the appropriate strength that they need for function, and therefore the appropriate transport of molecules along the neurons it's impaired, which then results in the degeneration of the axons, which then leads to the generation of muscle control. So in that regard, the neurofilaments are very important to maintain proper neuron function. The last intermediate filaments that I would like to introduce you to are shown here in the upper right. So what we have in this image is an epithelial cell, and this square it's showing you the intermediate area between the nucleus and the cytoplasm. So there you can see the double nuclear envelope, a nuclear pore, and right underneath it you have a set of blue lines, and those blue lines are going to be the nuclear lamina. The nuclear lamina, it's shown over here in this electron micrograph. It's shown to be a mesh of nuclear laminin, which is going to generate the structure below the nuclear envelope, and therefore gives structure to the nucleus. During cell division, when the nucleus is disassembled, the mesh of laminin is going to be phosphorylated to break it down, and that is going to be important in the division of the nucleus to allow the chromosomes to be segregated later during cell division. So now that you have having an introduction of the intermediate filaments, let's take a moment to look at some of the cytoskeletal functions that are available for all three of the molecules that we have discussed: the cytoskeleton, actin filaments, microtubules, and intermediate filaments. So here this cell, looking at a keratinocyte, it's showing you different functions that you can see for intermediate filaments and actin filaments. In number one, what you can see at the leading edge of the cell, keratinocytes can move sort of like a spaceship. This one will be moving towards the left-hand side. You see the branching of filament networks, which are extending and then exercising force at the edge of the membrane. That is going to move the membrane forward to allow for the appropriate movement of the cell. So you have an interesting movement of actin dynamics where the membrane, the actin network is being polymerized underneath the membrane, and therefore with the ARP complex, they undergo branching that can then put a force forward under the membrane to allow the cell to move forward. In number three, we have actin filaments which are crossing the cell, and there you have myosin motors which are bound to vesicles, and they are using their ATP hydrolysis to move the vesicles across the cell. In the nucleus, shown over here as this intermediate blue lines, that is the laminin network, which is containing the nucleus in its shape. So, last but not least, here number 5, you have a series of intermediate filaments and actin filaments that are used to mediate cell adhesion. So the actin dynamics are going to be able to induce membrane extension for movement. The filamentous network that is generating the bundles is going to give the cell structure. The myosin motors are going to help in contracting and vesicle transport, so contracting the membrane, for example, at the lagging end, or also helping to move vesicles across. The actin bundles and the intermediate filaments could play a role in cell adhesion, and the laminin network around the nucleus plays a role in nuclear structure. So here we have an epithelial cell, and this epithelial cell, as you can see from the membrane here in C, the membrane is undergoing different conformations. Those conformations are going to be required for the cell as it is exploring its environment and gathering information. So right below the membranes, you have the membrane, the cytoskeletal cortex, and that cytoskeletal cortex is made of actin filaments, which is spreading throughout the entire cell, giving the cell structure and shape. At the leading edge of the cell, where the cell is moving, you're going to have lamellopodia, and the lamellopodia are going to include proteins that can bind to the actin filaments, and therefore, as you can see by the arrows indicated over here, they are able to understand and translate the force of the cytoskeleton at the leading edge of the cell to push the membrane from underneath it forward. The phylopodiums are the extensions of the membrane that go forward, and again, membrane actins are going to be working together to extend the membrane of the cell in the direction of the phylopodiums. But the cell is not the only organism that can benefit from polymerizing actin. As mentioned to you a moment ago, the polymerization of actin exerts force that can push the membrane forward. So, in a sense, any particle that is going to induce actin polymerization on it can then receive that force and therefore be pushed by the actin polymerization forward. And this is what the bacterium Listeria monocytogenes have done to propel itself inside a host cell. So this bacterium can infect a host cell through a mechanism of phagocytosis, and then is able to escape the phagosome and therefore be free inside the cytoplasm. But at the pole of the bacteria, the microorganism is able to organize an actin filament to make it to grow. As the actin filaments begin to polymerize at the edge of the organism is going to now be pushed by the actin filament. What you can see here in the left is this explanation that is seeing in this cell has been stained to show actin. So what you can see is the actin ruffle that is going all around the cell, so that's the network around the cortex. And you can also appreciate some actin that is spreading across the cell as part of the cytoskeleton. But these little red structures inside the cell are the bacteria, and behind the bacteria you have what it seems to be an active tail, and that actin tail looks like a comet tail. So the microorganism in the interface of the organism and the actin tail is having the polymerization of actin, and that polymerization of actin is able to exert a force in the organism to propel it forward. The farther away you go from the position where the organism is having the actin polymerization happen, you now have the actin tail to begin to disintegrate, and that you can appreciate at the end of the tail. So in this one here that I'm showing at the bottom of the cell, you have the microorganism in red. You have a nice actin tail that is beginning very strong at the lagging end of the bacteria, and then it extends several bacterial lengths back until it begins to disintegrate as the actin polymerization begins to unravel and it begins to depolarize. But it is, as you can see, it is brightest in the interface of the bacteria and the actin nucleation side, because the bacteria has piggyback and hijack some of the actin nucleation mechanisms, and therefore it's able to use the nucleation of actin to propel itself inside the bacteria. So what I'm going to do is to show you this video. Oh crap, I cannot show you the video, but what I would like you to do, click on this video screen down here, and you will be able then to see how quickly the Listeria myelocytogenes is moving within a cell when you visualize it in vivo. So now, not only is Listeria able to use actin as a way of movement, the cell itself is able to use actin as a way of movement, and it is adhering to a substratum. So what we appreciate of here is that you have, for example, a glass slide. Here is our cell. This is an eukaryotic cell that is able to have actin underneath the membrane. So this is here shown as the orange actin cortex underneath the cell membrane. So now you have the lamellopodia, this leading edge extension of the membrane, where you have polymerization of the actin cortex right at the leading edge of the cell. What you have, because of that actin polymerization, it's the membrane being pushed forward, and that is shown here as this red extension of the actin cortex. At the same time, the cell is making contacts with the substratum, and therefore the cell can attach itself at the bottom, but allows the membrane to move forward. At the lagging end of the cell, you have myosin II motors, which are pulling at the actin cortex, bringing back the tail of the cell in a contraction mechanism. And therefore, as you can appreciate, as the leading edge in the lamellopodia is extending forward, the lagging end of the cell is being pulled back by contraction. So this interplay between the leading edge and the contraction is going to allow this eukaryotic cell to move forward in the direction that it needs to go. So this is what is happening at the leading edge of the cell. Here in gray we have the plasma membrane, this line over there, and that is the leading edge of the cell. The arrows in blue indicate the direction of movement. We went in class on Wednesday and we learned about the ARP complex. This is a protein that is binding to the minus end of the actin filament, but also it can bind within an actin filament, allowing it to branch. So it also helps with the polymerization of actin. So at the front end, at the leading end of the cell, what you have are complexes of ARP that are binding and therefore generating allowing the nucleation of actin filaments forward, and those are going to interact directly underneath the membrane of the cell, pushing it in the direction of the polymerization. What you also can appreciate from here is that at the end, you can begin to have the depolymerization of the actin macrofilaments, generating more actin that could then be exchanged. The ADP on it could be exchanged for GTP, that then allows it for be recycled and be used again for the polymerization of actin. We also have a capping protein that is binding to the plus end of the actin filament, and that prevents it from growing anymore. Eventually, only the microfilaments that are growing are the ones which are able to exert force underneath the membrane to facilitate cell movement. Now, another one of the proteins that I would like you to remember is myosin 1. And myosin 1, as we discussed, it is a simple motor myosin that works similar to a kinesin. It's actually related to kinesin in some ways, not to dynein directly, but to kinesin. So as a molecule is able to always move towards the positive end of the microtubule. So here, what we have now is a vesicle. That vesicle has myosin 1 bound to it. And because of the myosin 1 is able to hydrolyze ATP to move along the actin filament, it's able to move towards the plus end of the filament, which in this case is capped underneath the membrane, and therefore move this little small vesicle towards the plus end. At the same token, the myosin protein could be bound at the membrane itself, and then when it binds to the microtubules, since the myosin motor itself it's moving towards the positive end of the microtubule, a free microtubule like the one shown here in C is then being displaced to the left side. And this is something that we can observe readily in the laboratory under the microscope. So again, myosin 1 is able to move vesicles towards the plus end of the actin filament, or is able to then move filaments themselves from one side of the cell to another one as it is pushing the myosin, as it is trying to reach the plus end, but because they're tethered to the membrane, they don't move, they only move the filament. Now, in this image, now let's take a look at the microtubule network. And in this microtubule network, what we have, it's the showing in here in green, in the cell on the left, the microtubule organizing center, which is sprouting microtubules outside here in green. The ER is shown here in light blue, and the nucleus also with the nuclear membrane, which shares the ER. Nucleus is here in brown. And the Golgi apparatus, it's nicely kept around the nucleus. And that is all done by the tug of war that the ER and the Golgi are going to be receiving from the Dynein and Kinesin molecules. So in the first part, we have endoplasmic reticular staining. So what you can see when you compare the endoplasmic reticular staining is that it is superimposed nicely on the microtubule network. So the ER is being pulled outwardly along macrotubules by kinesins, so it is extending the endoplasmic reticulum all the way across in very beautiful blue strands along the microtubule network. On the other hand, you want to maintain the Golgi apparatus close back to the nucleus. So by having the Golgi apparatus being pulled by Dynein molecules, now the Golgi apparatus shown over here can be seen that is being brought all the way close to the microtubule organizing center. So in that way, you can keep the compartmentalization of the cell by organizing it along the binding of kinesins to pull it away from the microtubule organizing center, or the binding of Dynein's to bring it close to the microtubule organizing center. So here in yellow, you have the Golgi stain. In light blue, you have the ER stain. And below, in the two images, in green, you have two different cells. One, the microtubule stains, and the other one also showing microtubule stains. So the ER staining on top corresponds to the microtubule staining on bottom, and the Golgi staining on top here corresponds to the Golgi staining here at the bottom. So what? One cell, one ER and microtubule stain, and cell number 2, the Golgi apparatus and the microtubule staining. Microtubules are also extremely important in cell movement. And what I want to introduce you is to the structure that is at the core of the flagellum and cilium movement, the axoneme. The axoneme, it is a complex microtubule array of 9 doublets and 2 singlets. And because of that, we call it the 9+2 array. What you can see here on the left is an electron micrograph of the axoneme. And as I labeled for you the doublets, shown here from 1 all the way down to 9, and I also put in green the label of the single microtubules in the middle. Now, the cartoon on the right is showing you the actual structure of the axoneme. So, in a doublet, over here, what you can have is one entire microtubule with all 13 different protofilaments, and we tend to call that the microtubule A. Attached to that, it's a microtubule called B, and together these 2 microtubules form the doublet. Now, each doublet is bound to one another by a molecule called nexin, and therefore all 9 doublets are tethered together with nexin bridges. The other molecule that it is important to have are the radial spokes. The radial spokes are emanating from the A tubule within the microtubule doublet, and they are interacting with the inner sheath that is surrounding the inner central microtubules. So now, as you can appreciate from this image, you have 9 different radial spokes that are interacting with the inner sheath where their 2 central microtubules are present. And each of the 9 microtubule doublets are linked together by nexin molecules. Now, to the A microtubule of a doublet, you have covalently bound Dynein motors. And what you can appreciate is that those covalently bound Dynein motors are going to be able to interact with the neighboring doublet microtubule, usually only with the B portion of the doublet microtubule neighbor, and that the other microtubule, the A component that has the Dynein molecules, is able to interact with the next B subunit of the neighboring microtubule. And that is happening all around all the 9 different microtubule pairs that are around the axoneme. Also important to know, it's that the entire axoneme structure is covered by the plasma membrane. So the entire axoneme and the flagella and the cilia are internal organelles. And I would like you to think about why that may be the case. So I will leave that question there for you. Now, in this slide, I have the architecture of an entire cilia, and that cilia, is, as you remember from the book, is composed of a basal body, and then you have the 9+2 axoneme, and at the top you have what is called the ciliary cap. Now, at the top of the ciliary cap is where we have the plus N of the microtubules. All the microtubules present in the axoneme, the 9+2 axoneme, has an N in this plus cap. The axoneme, it's the inner structure of the cilia or the flagella. Both of them have them. And as I mentioned to you earlier, they have the A microtubule and the B microtubule. So the Dyneins, which are bound to the A tubule of the doublet, are going to use the B tubule of the neighboring doublet to move along the axis. What we'll see is that at the bottom of the cilia, shown here a little bit more inside the cytoplasm, it's going to be the basal body. And the basal body, as we've discussed, it is a specialized centriole. But this specialized centriole has triplets. So it has 9 different microtubule triplets containing A, B, and C tubules. And those are together organized in such a way that are going to keep the structure. So you can see it here in number 3. You have the A, the B, and the C microtubules. The B and the C are partial microtubule tubules in the entire microtubule network, and that forms the basal body. And from that basal body, it's where you now begin the nucleation and architectural composition of the entire axoneme structure. And the axoneme structure is the one that is going to generate movement. So how that is happening? So here we're looking at two doublets, and these two doublets are attached together by the Dynein molecules that are present on the A microtubule in the left, and they are using the B tubule on the microtubule on the right as a track to move. When you add ATP, which is required for Dynein to move, what the microtubule Dynein are going to do is to move towards the negative end. In this case, because these two microtubules are not tethered to one another, one microtubule will be displaced to the bottom and the other microtubule will be displaced to the top. And this is where the nexing molecules come into play. When now the microtubules in the axoneme are bound together by nexin, you only have a certain amount of give when the acting molecule begins to when the Dynein molecule begins to exercise its strength to move the microtubule. So what that is going to result, it is on the bending of the two different microtubules instead of the sliding of the microtubules away from one another. The Dyneins are going to try to move towards the negative pole, but because you have the nexing protein linkers, they're only able to bend. When they're bending, what you have now, it's 9 different microtubule structures that are bending to one another, and that is what is going to generate the undulating movement of the cilia and the flagella. So in this image, in the left, what we have is pulmonary flagella that is used to eliminate a lot of the sputum that is present and mucus that is present on the lungs of people. And what we have to the right, it's the two different kinds of movements that the cilia are going to organize together and exert to move. One of them is going to be the power stroke, and the power stroke comes from the left and moves the microtubule all the way to the right. But when it is all the way to the right, it's going to coil and link by itself, moving all the way back to the initial position of the power stroke, and that is called the recovery stroke. So every time that you have a power stroke that is going to allow the cilia to move from left to right, then you have a recovery stroke that is going to recock the cilium from right to left. And the combination of the power stroke and the recovery stroke help the microtubules, help the cilia move forward. This last image that I want to show you was one of the photographs that was submitted and awarded 8th place in the Nikon Small World 2012 competition. And this photograph that came from Gerd Gunther in Germany, it's looking at the cilia in the sea gooseberry larvae using differential interfering contrast microscopy. So because of that, what you can see now are the cilia as they are going through the recovery stroke. So the one on the right is becoming too cock, and as you can see from the one in the far right to the other ones in the left, as they're moving, you're beginning to appreciate how the cilium is becoming more and more and more straight, to eventually together all of them move to the right again to exert the power stroke movement to allow then the larvae to move. So that will be the last image that I want to show you today. That concludes the lectures that we're having about intermediate filaments and as well as cytoskeleton. And then I will see you on Friday to continue and have our last flip lecture. Have an excellent day, and I'll see you tomorrow.